So what we asked our four distinguished panelists to come in and talk about is what's their take on evolution in a very local setting, in their backyard. And allow me to gush over our four panelists. Cole got here first, so he's going to go first. He's associate professor in the Department of Entomology at Cornell. He received his undergrad at Washington University and his master's and his PhD at the University of Kansas. And he focuses on behavior in arthropods in his research. Willie Bemis is professor in ecology and evolutionary biology here at Cornell and former director of the Shoals Marine Lab. He received his undergrad at Cornell and his master's from the University of Michigan and his PhD from UC Berkeley. His research focuses on anatomy, development, evolution of fossil and living fishes such as sharks, lungfishes, coelacanths, bowfins, sturgeons, and paddlefishes. Jeremy Searle is professor in the same department, ecology and evolutionary biology. He received his undergrad from Oxford. I learned something in preparing this, by the way, that you can say oxen. I did not know that. So if you are an undergraduate from Oxford, you can say oxen. Got his undergraduate from Oxford and his PhD from the University of Aberdeen. His research focuses on the evolution of mammals, especially speciation and chromosomes in small mammals. And Brooks Minor is assistant professor in biology at Ithaca College, received his undergrad and his PhD from the University of Washington, and he is a freshwater ecologist and evolutionary biologist.
Well, I took the charge of talking about things in the backyard literally as far as the key example. This will be a very short talk, not too many asides here. But I want to start by mentioning my time here as an undergraduate, which was a very exciting period for me. I showed up all eager to study marine invertebrates, and I ran into this guy named Harvey Poe who said, "Wouldn't you rather study vertebrates?" And I said, "Yes." And within my first week here as an undergraduate, he had me out at Ringwood Pond, which is one of the wonderful pieces of the Cornell Plantation's properties east of here, and had me out in the middle of that pond taking turtles out of turtle traps. It was monitoring all the turtles in Ringwood Pond. And I thought that was pretty cool, and I was pretty happy to be a herpetologist here. And I got very interested in our local herps. Thought for a long time I was going to spend my whole life studying herps, but no, not really. I ended up working on fishes. But as it happened, when I came back here in 2005, I bought a property that abuts on the Ringwood property. And so I have many of those same amphibians that we used to go up there and study when I was an undergraduate. So a couple of years ago, it's actually embarrassing, it's 7 years ago now, I went up to our pond up the back end of our property there, and we had a little rubber inflatable boat for my son, and it had been left out over the winter, which was not a good idea. But I flipped it over, and underneath it, I saw a spotted salamander. But actually, there wasn't just one. There were 4 of them in that group. There's 5 and 6, 7 and 9, 10 and 11, 12, 14. So there were 14 spotted salamanders. You can see them, some of them, more or less, in amongst the mud and detritus here. And they were right next to, thank you, they were right next to, there we go, put it that way, this whole display of the salamanders that have been underneath the boat is about 2 feet away from the edge of my pond. And they were gathering there. These are males and females, and they were getting ready to go into the pond to have their spawning activities. Now, this is going to take place around here, as most of you know, just in a few weeks. And it's always very exciting up by us because Kelly Zamudio maintains the tunnels under Ringwood Road so the salamanders can cross underneath and not be out on the street as they were when I was a young person. We have similar tunnels there in Amherst, where I taught at UMass for many, many years. At any rate, I love spotted salamanders. I've been fascinated with this group since I was a kid. And I take the notion of backyard and spotted salamanders to a much larger worldview eventually. And I'll just do that for you in a couple of slides. This is a picture of a larval tiger salamander. It's another member of the genus Ambystoma. There are about 30 species alive, or living species in the genus Ambystoma. And you can see a number of really cool things about this that you probably already know very well. But they have external gills as larvae. They have a very prominent tail fin. The head is pretty flattened. These characters don't change at all at metamorphosis, 4 digits in the forelimb and 5 digits in the hind limb. But at metamorphosis, many of those other features do change. They lose their external gills. The head, though it's still flat, becomes a little deeper. The eyes have much more prominence. They lose their tail fin, and they come out and they migrate around on land as terrestrial adults. Now, the thing about amphistomatid salamanders is that these 30 species are distributed in North America, but most of their diversity is actually pretty far away from here in New York State. Much of their diversity is in Mexico. And for my purposes, Mexico is still sort of the backyard. I spent a lot of time down there when I was younger, and one of the most amazing salamanders in Mexico is this guy. This is Ambystoma dunarilii. It comes from Lake Pátzcuaro in Michoacan, west of Mexico City. And it's the only place in the world that this salamander occurs. And you look at this guy, for a long time, people gave it its own genus. They said, we're going to call this thing so different from other amphistomatids, we're going to call it Bathycyrodon. So I tend to still call it Bathycyrodon. And when I was a kid, we called them Bathies because they were so cool. Just the biggest, most exciting salamanders you can imagine. They eat them there in Pátzcuaro. They're fished by the local people. They sell them in the market. That's where we would buy our specimens when I was down there. And these guys are perennibranchiate. They retain these gills, the external gills, throughout life. They have a flattened head. You can't see their tail fin, but they keep that too. And they never metamorphose in the Lake Pátzcuaro situation. If you bring these things from Lake Pátzcuaro back to Ithaca and spawn them with other members of the genus Ambystoma, you can get metamorphosis to occur. Or if you supply hormones, such as thyroxine, to the water, you can convince some of them to metamorphose. But they never do in nature. And so this wonderful pattern of retaining these juvenile features throughout life is pedomorphosis, of course, and it's the retention of juvenile or larval features into adulthood. It's important in the evolution of many, many groups. And it turns out that the amphistomatid salamanders, not just our spotted salamander and our friend Bathycyrodon, but in particular another amphistomatid from Mexico, Ambystoma mexicanum, commonly known as the axolotl, are the classic examples for one type of pedomorphosis. This type of pedomorphosis is the deceleration in the growth and rate of shape development. And essentially what's happening in pedomorphosis by deceleration is the rate of development is slowed. That's the Ka versus Kd, the descendant rate of development is slowed. And this was the inspiration for a wonderful paper co-authored by Steve Gould and Per Alberg, George Oster, and Dave Wake, published in the early 1980s in Paleontology, to look for other types of pedomorphic events and how you can model those. This is the second type. This is hypomorphosis, and in this one, you just simply stop development, keep the same rate, but you stop it early. So you wind up with a descendant that has a very different shape than its ancestor. And then this is the third possibility pointed out by those wonderful authors in their paper in the early 1980s, in which case we've just postponed the start of the development of shape. Now that sounds like a long way away from backyards, but from my perspective, thinking about the time that I spent here as an undergraduate, when we were raising axolotls in Stimson Hall, and thinking about these issues in pedomorphosis before we had quite the nice model that's presented here, it's very backyard for me. And I love that time I spent in Mexico with Brad Schaefer and others, exploring these beautiful lakes high up in the Mexican volcanic belt. So that's what I needed to say today. Thank you very much.
So, thanks for the invitation, Warren. And the idea that Warren put on us, evolution in your backyard, I took a little bit more literally than Willie did, and So this is kind of things that we can see in my backyard. My wife Linda Ray and I live over the other side of Trumansburg, out in the woods there. And so I just put a picture of Darwin's yard and his backyard. Ours is a little bit different than that, not nearly so groomed as that. But I want to talk about two stories. I'll be glad to talk to you about insects, but I was thinking about interesting things, and we have there slugs and salamanders that has been on my mind lately, and then woodpeckers. And so here's roughly what my backyard looks like. This actually is sapsucker woods, but we have a nice woodlot there. And if you go out and turn over one of these logs that you might find in the yard, you can see these creatures, slug and salamanders, especially redback salamanders are quite common around here. And they're in this same kind of wet habitat, but the way that they get there evolutionarily is interesting to me. I've been thinking about it a little bit lately. So let's start with the slugs. And here's a tree of the mollusks, and there's some groups down here that are not so important except the systematous, but most of you probably know the cephalopods, octopus, squid, nautiluses, scaphopods, clams, and then we've got gastropods up here, snails and slugs. And they're mostly marine or freshwater, but several times within the gastropods they become terrestrial, and the slugs are one of the groups that do this. They still like really moist habitats. And so they'll, the bodies are moist, and they'll leave this kind of slimy trail walking around. You can find them. The eggs are little spheres, a little cluster might be under the log there. You lift the bark and you can see it there. Some of you might see them in your garden there, but we see them in the woods, and the little larvae are developing in the eggs. Here's one coming out of an egg. It looks really cute. He's got his ocular tentacles, his kind of olfactory tentacles coming out of the egg. But one of the really interesting things about these creatures that you notice when you see them, they're out grazing, eating fungi, algae. This leopard slug will eat another slug. Is this big hole here on the side? And this is the pneumostome. It's a breathing hole. The ancestors of these creatures, most of those other folks on the cladogram, have gills for their respiration. And these terrestrial ones have gotten rid of their gills, and they've developed a lung to get in there. They're sometimes referred to as pulmonates. And here's a shot. It's not open all the time, but here's the pneumostome here, and this is a little, there it is, opening, 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 closing, closing. So it's taking a breath. It actually opens that up, takes air into the lung, closes it up, presses the floor against that, and the inside is highly vascularized. Here's the interior of the lung. This is a transmission electron micrograph. The capillaries run around the side there to do gas exchange. So this creature has developed a lung to be in this terrestrial habitat. As long as it's moist and the tissue is moist, gases can move across that very easily, just as they do in our lungs, and it makes its way around in that habitat. What about salamanders? So Willie talked about salamanders. We didn't coordinate this. I'm glad I put a spot in
that's great.
one there for you. Here's some of the families that we have around here. The newt, the F stage of the eastern newt, and you might see these and sometimes oranger than that on the trail. We've got seen these occasionally in the pond in the backyard. We don't have hellbenders in our backyard. I did grow up in Missouri, eastern Missouri, and we've got an Ozark hellbender there. There is an Allegheny hellbender in southwest New York, and then the amphistomatids that are in the York. These things all have lungs. Even the aquatic ones have lungs and they do respiration with them, although the hellbender may do quite a bit more through the skin. But let's look at sort of who's who among the salamanders then. A bunch of families that you don't need to know about, but here's the hellbenders here, the salamanders there, the hemistомatids, and then here's this group up here that I didn't mention, the Plethodontidae, and these are known as the lungless salamanders. And so under this log, you'll find a redback salamander, little spherical eggs. They don't lay their eggs in water, they're completely terrestrial. They stay in this moist habitat and will put their eggs there. They undergo direct development, and so here's the little salamanders coming out of the eggs there. And so we've got these two different groups, and with the slugs, they've evolved a lung to get into this habitat, to be terrestrial, and as long as it's moist, their lung is going to work, they can do gas exchange across there. These salamanders come from a group of creatures with lungs, and they've lost their lungs to be in this same habitat next to these other creatures. As long as it's moist enough, they can take enough oxygen through their skin, which is highly vascularized, and exist that way. And it just struck me as totally weird that these two different groups, one gets lungs and it can last in that place, and another one loses lungs, and it's in that place. So here's a little conclusion. They've gotten into this same habitat from great diverse lineages. The slug evolved the lung to do this, and the salamander lost its lung, and it can still do this. All right? Second story. Back to the woods. Again, sapsucker woods. This is a nicer picture of woods than what I would take out in our woods. And one of the kind of a big principle of evolution is that distinct sister species can evolve when populations are separate and evolve reproductive isolation. Then when they get back together, they might avoid competition by exploiting different resources. This is sort of what we call the allopatric speciation model. It's the one for which we have the best evidence. There are other models, but I want to focus on this, how they become different, not just in terms of reproductive isolation, but in terms of exploiting different resources when they get back together to avoid competition. And of course, this is what Darwin's, most people will know about his evolutionary ideas, comes from looking at the finches, this array of birds with different beak types, and to exploit different sizes of seeds or insects in seeds, prey in different places in the habitat. And as Warren said, we can't all go to the Galapagos and see this there. It's a small group, it's really well known, but there is a group around here that allows us to see some of these same ideas, and we've got them in my backyard, and that we can talk about this character displacement. So the idea that the shape of the beak might change, so that characteristic can be displaced from one species to another, and the farther apart it is, the less competition there will be, and both could pack in and still live in the same environment. And so in my backyard, we've got little downy woodpeckers, and then we've got the next size up, the hares that are feeding there. We've got the red belly, and a little bit bigger than the hares, all doing basically the same thing. We live in a slightly lowland area, and we've got pileated in our woods, two pair that live in the woods that we know. They're all doing the same thing, but the pileated is gigantic. It makes a hole this size in tree trunks, and it can really get deep into stuff. The downy can't get in that far, but it can also exploit littler things like the goldenrod bald galls that have a nice little juicy maggot in there that's loaded with fat this time of year, and these guys can hang on there and eat that. So they're spreading out in the same way that finches do to be able to get in a habitat but not compete with each other for food. So there's not a really good phylogeny of woodpeckers yet. There are a couple of good ones, but they don't have all our species in them. So I put this together this is what we know about some of the relationships. So here are the guys. Those two are sisters, species, same genus, and they're like 50% different in size. There was an ecologist, G.E. Hutchinson, that said that's how packing should be. We call it Hutchinson's ratio. The pileated woodpecker and the red-bellied. But what about these other clades? Well, we have a couple more in our yard. The pileated and the northern flicker. So we don't get them so much in the woods, but we have a bunch of trees cut down where our septic field is. And so we have a grassy area, and these guys like to feed on the grass. So they're not competing with this bird. I mean, they're a little bit smaller than this, a few inches smaller, but they're not competing with them. They've gotten into a different habitat. And then if we look at the red-bellied, we actually have yellow-bellied sapsucker, and these guys are doing a different thing where they poke holes in the trees, same kind of behavior, but they're taking the sap rather than taking bugs out of there. So they're diverging that. So we'll finish with the conclusion from part 2, that they can respond to selective pressure by shifting characters to reduce competition, character displacement, and different birds with different sized beaks can eat insects at different depths. And the local woodpeckers are a great example. You don't have to go to the Galapagos to see this kind of radiation. Now, we don't know as much about the evolutionary history of these birds in this area as we do about the Galapagos finches, but you can go out in your backyard and see it, and it's just as cool. So I'll finish with that, and I'll be happy to take any questions.
Who wants to go next?
Good afternoon, everyone. Thank you, Warren, for inviting me. And I've only got one slide, and this relates to research I'm doing, which is sort of backyardish. It's about mammals that are very closely associated with humans, and maybe in the backyard, maybe in the house itself. So this is research relating to the house mouse. And I'm going to talk about what goes on with the house mouse in Europe, and then I'll say a little bit about what I think might be going on with the house mouse in Ithaca and around about. So the house mouse is quite an interesting species. It's associated, long been associated with humans. So essentially, with the development of agriculture and the first human settlements in the Near East, in the so-called Fertile Crescent around about 12,000 years ago, house mice were small rodents that lived on seeds, lived in rocky places, happened to be living in the Near East. So when humans started developing crops and cultivating them and storing them in structures, stone structures, that was kind of parent as far as house mice are concerned, because it was just having this wonderful food supply all in one place, and they could just go to town with that. And really, from there on after, they associated with humans, and there's good archaeological evidence that they did that, and we can see that they spread from the Fertile Crescent into Anatolia, which is the area of modern-day Turkey, where Turkey is in this sort of area here. and they spread into the human settlements there like Plethodontidae. But interestingly, they didn't really, you might have expected them to have spread into Europe proper with the Neolithic revolution, the spread of that through Europe, which was about 7,000 or 8,000 years ago. But it wasn't until about 3,000 years ago that we got archaeological evidence that they went into Western Europe. And that seems to be associated with the movement of, in the Mediterranean, a lot of maritime traffic that happened around like the late Bronze Age, Iron Age. And that was also the time when you got development of really quite large settlements. What seems to have happened was that humans were obviously being settled in Europe with the Neolithic revolution already 7,000 or 8,000 years ago. Those settlements were relatively small and they were exploited by native mice rather than house mice. House mice seem to need really quite large human settlements to be able to outcompete those native mice. And the native mice that were present in Europe and are still present in Europe are a species called Amphibians sylvaticus, which is what's called the wood mouse or the field mouse. And I haven't got a picture of that, but it actually looks incredibly like the deer mouse of North America, the Peromyscus, and has very similar habits. It's basically an outdoor living mouse, but is able to exploit human settlement. He's able to come into houses and does rather well when there are small human settlements. Anyway, 3,000 years ago, these house mice managed to spread into Western Europe. So that's really what we know about from an archaeological viewpoint. But the research that I've been doing is actually doing genetic studies on these house mice. And we've studied house mice throughout Europe using a particular region of the genome, and it's a rather strange part of the genome. Is we all obviously have chromosomes in our nucleus, and that's where the bulk of the DNA that we have is located. But we also have these small structures called mitochondria in our cells as well. And they have small circular sections of DNA, which is particularly easy to study. And so what we did is study one particular gene of that mitochondrial DNA, and we obtained sequences of that, and we studied that in mice throughout Europe. And then we used those sequences and we constructed a phylogenetic tree, and we've seen phylogenetic trees. Cole showed a couple. And that's those are kind of species trees, looking at the relationship of the species. But you can also look at the relationships of genes, sequences for a particular gene, which you can call alleles, different sequences. And so this is a kind of allele tree for this particular gene of mitochondrial DNA. And in the same way as you can see particular lineages which may be bringing together closely related species, you can see different lineages which are bringing together closely related alleles. Anyway, what we've done here is think about these different lineages, and we've coloured the different lineages separately, and we've given them different names. So they're between lineage A all the way to lineage F. And then what we've done is we've actually mapped out the distribution of these different lineages. And it ended up really very interesting, because we've got lineage A here that is associated with the Near East and gets into Mediterranean areas in the sort of eastern part of the Mediterranean. And we get rather a similar pattern with lineage B, except that manages to get out into Western Iberia as well, and out onto the Azores kind of lineages that seem to have been moved to, basically any mice that we see in Europe has got there not on their own legs but through human transport. So it seems that sort of Mediterranean transport by the Phoenicians or other people around this late Bronze Age, early Iron Age period has moved them around to this particular area. And then we've got this other lineage, C, which likewise seems to have a good Mediterranean distribution, but has this interesting distribution through Central Europe. And our take on that is that this is a lineage that was likewise spread around the Mediterranean, but actually started being taken by Iron Age people up through Central Europe. And it's well known that the Rhone Valley, I mean, people travelled around into sort of Central Europe using river systems, and the Rhone was very important. So we sort of suggest that it may have been using the Rhone that they managed to penetrate. Anyway, that's lineage C. And then we've got another lineage D, which is widely distributed in Scandinavia, also some indication in the Mediterranean. So this one seems to have got a bit further. It seems to have got through Central Europe, but also got up into Scandinavia. So that, we believe, again, may have been a transport through Central Europe. So after D, we've got E. And this is interesting because this isn't well represented in Europe, but it's very well represented in the British Isles and nearby areas of continental Europe. And what our suggestion may be for that particular lineage, all these lineages had to somehow have come from the Eastern Mediterranean. But what we believe here is that this was possibly a rather rare lineage that managed to get as far as the British Isles and nearby areas, and it just basically, that was the lineage that became common in the British Isles. And we know that the Phoenicians had sort of trade links through the Mediterranean leading up the coast of Iberia and possibly as far as the sort of tin mining areas in Britain. The last lineage is Clade F, which has similar characteristics. It's not represented at all in the Mediterranean, and yet has got this strong signal along the northwest of British Isles and western British Isles, along the coast of Norway and Iceland. So again, that somehow must have managed to penetrate that area and then was moved around these sort of Atlantic areas, and that has to be through seafaring that happened. So this is all quite surprising in a way. You know, humans travel all over the place. You would expect mice to be associated with foodstuffs that humans transport. And so in some ways you might have expected a real homogeneous mess of all different genetic types, because mice would be taken from one place to another and just sort of all over the place. But we actually have this very striking patterning in the genetics of mice, which is really quite interesting. And this, we believe, is related to the fact that we're looking at mitochondrial DNA, DNA associated with mitochondria. Because this is associated with an organelle that we find in each cell, this has got a different type of inheritance from our nuclear genes, the ones that we find in the standard nucleus. And our mitochondria are actually entirely inherited from our mothers, because basically the way we inherit our mitochondria is that they're present in the egg. You don't get any mitochondria from the sperm. And so what we think is happening is that this is showing us female history of house mice. And what we believe is that when female house mice colonize a particular area, it's actually difficult for later female house mice to displace them. So this is showing us first colonization of these areas by female house mice, and therefore that you can see that pattern could be really set in place quite early on and difficult to disrupt by later movements. We've got various evidence to support that. Okay, so that's what's going on in Europe. So what do we think might we see in terms of these lineages in upstate New York? Well, what we've got to think about is house mice are being moved around by humans, and therefore we have to think what humans would have moved house mice from Europe to North America. And the prediction I would make is that they would be, we know that there's a lot of association with the New England area and New York as well. There's kind of associations with either the British Isles or the Netherlands or places close to us, Northern Europe. And although the Spanish clearly had impact in southern areas like Florida within the United States, they didn't have much impact up here. So my prediction with the house mice in upstate New York is that they're either going to be this lineage, E, which is the most widely distributed lineage in the British Isles and nearby areas, including the Netherlands, or else F, which is also widely distributed in the western part of the British Isles. So I think what I actually haven't, even though I've been in Cornell and Ithaca for the last 4.5 years, I still haven't got any house mice no donations of house mice from Ithaca. You can get this picture. Okay.
Tonight.
It's far more common to get these guys, Paraniscus, because anybody living sort of in a sort of wooded close to a wooded area like Cole, the mice that will come in. So what you've got to be careful about is if you've got a mouse which has got large ears, large eyes and a very white belly, that's not the sort of mouse I'm interested in. This is the sort of mouse that gives you Lyme disease and hantavirus. I really don't want those. These guys give you things like leptospirosis, but I can deal with that much more clearly. They've got smaller eyes, smaller ears, they're very grey, and their belly is grey, they just look grey, these guys look brown on the back. So I'm interested, I'm actually curator of mammals at the Cornell University Museum of Vertebrates, and we really would like to expand our house mouse collection. And so we want to see which one of these lineages is in upstate New York. The interesting thing about, so this lineage lineage E, I believe is a marker for sort of Iron Age movements between Britain and continental Europe, and we can actually use further studies on this to actually understand the history of Iron Age people. And so we're sort of thinking ways of exploiting that. This one is this sort of maritime distribution. We are convinced relates to Vikings, and that they were spread around. This is very much the realm of influence of the Norwegian Vikings, including colonisation of Iceland. And we've actually got Viking Age mice that we've managed to study from the archaeological sites in Greenland as the eastern and western settlement, and we've shown that they're of this lineage. So in terms of Viking penetration or Viking interactions with mainland North America, this lineage of mouse is quite interesting. But that's, so this is kind of, we can see that the evolution of the mice in a sense has happened a while ago, generating these different lineages, and what we're seeing now in terms of your backyard in Ithaca is the consequences of that.
First of all, you have a slide up there, E and F. is it possible, and I'm a non-biologist, so I'm not sure I know how to ask the question, but
Okay.
it looks like ENF could be mutations because they don't have representation back in the Middle East, as opposed to some kind of a species differentiation as they migrated out.
Yes, I mean, they've got to, you know, they've got to derive from something before, you know, getting to that lineage. And basically the degree of, all of these seem to derive from some sort of common ancestral type that was in the Middle East, and we don't have a strong indication from this phylogenetic tree that they derived at a different time. So we think that all of them were actually present and they just haven't been represented. And I think it could be just chance that we have rarer types that have managed to get out of from this eastern Mediterranean area.
What time frame are you talking about between A and F?
Well, I think all of, as I said, I think all of them were already present at the time of association with, early association with humans, and we're just, what we're seeing now is those genetic forms, in a sense, that have been sort of separated out and have ended up in different places.
And if you want contributions of house mice, do you want them dead or alive?
Dead, please.
Frozen okay, or just?
For any, I mean, I can give you, maybe the best is just to put on my email address. Is that the easiest for people?
Maybe the kind of not knowing visually the difference between a house mouse and a deer mouse, the kind that are going after the peanut butter in my mouse traps.
I mean, a lot of people are going to have deer mice. I mean, the thing about house mice is they're the ones that sort of you get all year round, living in kitchens, you know, just, they often can achieve really quite high densities. The deer mice will never achieve high
And they're
densities.
really gray.
And they're really grey.
Mine are really great.
Yeah, that's the difference. If you see that white belly, it's not a house mouse. But we're very interested, and basically, I think probably what I would suggest to people have them is sort of leave them in the museum, because I think it's useful to have. So I mean, if people snap track them and they're not in terribly good condition, I don't know what people feel about that, but actually cutting off a small piece of ear is all we need, because we just need DNA essentially. So it's yeah. Great.
Sure.
Oh no, frozen birds, yeah, they go over here. Bring you an office mouse.
Office mouse.
Yeah.
I've never seen this, so I don't know if they're this, but I highly doubt it. Because they're,
So as long as they're not escaped lab mice.
no, because there isn't anybody who does.
Office mice have carpal tunnel syndrome, is that?
I don't know.
Yeah, no, I'd be really.
Yeah, no, I've definitely learned in the office that I've been moved into since last August that all food left in my office must be in a closed container. So there's definitely some that
It's not the custodian?
no, because there's fecal evidence as well.
Guy comes by our building monthly to check and see if we have had any bugs. And we could talk to him, he's a really nice guy, very interested. So we'll check that guy.
Yeah, that'd be great. Yeah, that'd be great.
So Warren, thanks for the invitation. Thanks to you guys for being here. As I was sitting here during that earlier presentations, I started to think about how I had the least Darwin-like phenotype among the four presenters because I don't have a gray beard. But then I started to think about how the Darwin phenotype that we see, like on the poster with the long gray beard, is just Darwin at a certain stage in his life. And there's actually a photo, and if I had thought of this in advance, I would have put it on here. But I didn't. There's a portrait of Darwin from when he returned from the voyage of the Beagle. And not only does he look younger, but as I was sitting there mulling over this visual thing, I remembered that picture, he has these like huge sideburns that are not totally unlike the ones that I'm sporting here. So I figured that fit in just fine. So what I want to talk about today has to do with not something I can observe in my backyard because I don't have a pond or a lake in my actual backyard. But we do have a lot of lakes and ponds around Ithaca. And I started to think about what there is that one can observe about a lake or a pond over the course of, for example, the spring to summer seasonal change that represents an important evolutionary event. And the one that I'm going to tell you a little bit about today, it's evolution happens on a lot of different time scales. And in terms of the longer time scale, differences among species, lungs versus breathing through the skin, like what Cole talked about, those sort of macroevolutionary patterns, is one important element. There are other evolutionary events and processes that occur repeatedly, every year, over and over in sort of this cycle that occurs because of the nature of seasons as we get them in a place like Ithaca. So that's kind of what I wanted to focus on today. So this is a graph. Was there a pointer? I'm just going to bring mine up and I forgot to grab it. Thanks. So in this graph, when we think about a lake or pond habitat, you often start to see a lot of graphs that look upside down. And that's because whenever you put anything on a depth axis, it makes more sense to have the y-axis going down to, for example, 7 meters deep in the water, rather than to represent 7 meters going up. So on this axis is the depth in the water at which a circular disc that looks about like a dinner plate, it's about that size and shape, when you lower it over the side of a boat, it's a white dinner plate type thing, it's called a Secchi disc. When you stick it in the water over the side of the boat on the rope, you can see it. And as you lower it deeper and deeper in the water, eventually you can't see it anymore. And that depth at which you can no longer see the Secchi disc is known as the Secchi depth. And it's a really reliable, even though the device has been around for over 100 years, it's a really reliable and very simple way of measuring how clear the water is in a particular body of water at a particular time you take the measurement. And when you sort of stop being able to see the disc, actually turns out to be not very much influenced by whether you do it on a sunny day or a cloudy day, or whether you do it when the sun is high in the sky or low in the sky. It is almost entirely determined by how clear the water is. So if you lower this disc over the side of a boat, so I put this blue oval here to represent the surface of the lake. And if you were in a boat on the lake and you lowered this disc over the side and you measured the depth at which you could no longer see it, and you were in a north temperate lake, like most of the northern US, and you did this over the course of a year, the exception of when you couldn't do it because it's covered in ice, you would probably see a pattern that looks something like this. So when the dots are closer to the surface of the lake, it means that the point at which you can no longer see the disk is closer to the surface. So it means the water's cloudier, there's more stuff in the water, and it's not as crystal clear. When you see a dot down here, it's something like 6 meters, that's really clear water. That's 6 meters down and you can still see barely a white disk about the size of a dinner plate. And what's distinctive about this particular example that I'm showing you is there's a general pattern that's predominant among north temperate lakes of this period in the late spring, early summer where the water gets really clear, but it doesn't last, and then it goes back to not being as clear anymore. And I point this out because you don't even necessarily need a boat or a Secchi disc to observe this. If you have a lake or pond that you visit frequently for whatever reason, for me, even not for doing research, one of the things I've learned since moving to Ithaca is that it's really fantastic to go swimming in some of our local lakes and ponds, and there's a whole water temperature thing that goes along with when it's a feeling to go swimming as well. I won't get into that. But this period here is pretty distinctive, and it has a name. It's called the clear water phase. And it has a really interesting cause. What happens is, in the spring, once there starts to be warmer water, ice melts away if there was ice, and longer days, so more sunlight. Basically, you start to get the same kind of thing that we see in the terrestrial landscape in the spring. You start to get a lot of plant growth. And in an aquatic ecosystem, plant growth means algae, which are little single-celled microscopic organisms. And those algae are one of the main contributors to the lack of water clarity in a lake. So that's why if you ever happen to be up in a high alpine system where there's very few nutrients and a very short growing season, you get crystal clear water. Algae isn't the only thing that determines the transparency of water, but it's a predominant one. And what's going on in this clear water phase is you get this thing that happens in early spring where there's sort of a bump in sort of how much stuff there is in the water. That's because of what's known as the spring bloom, and that's a whole bunch of algae growing and reproducing because it's finally warm enough and there's nutrients available and there's finally longer days in sunlight. And the reason that they all disappear, not completely, but the water gets a lot more clear, is because they're getting eaten. And one of the predominant consumers of algae in north temperate lakes are species belonging to the genus Daphnia, which has the common name water flea. They're little crustaceans, they're not actually fleas, they're not insects. And they're about 2 millimeters in size, so these aren't, although their food is microscopic, they're not microscopic. And we grow them in the lab and we can see them with the naked eye. We put them under the scope to take cool pictures like this. And they're filter feeders. They're constantly sucking in water and moving it across all these little combs they have with these fine hairs and collecting particles out of the water and consuming it. And this example of 3 different years, 77, 81, and 98, this is from Oneida Lake, which is up near Syracuse. And what you can see here is, now I'm not graphing the Secchi depth or the water transparency, but there's a green dashed line for the concentration or how much biomass of algae there is in the water. And then there's a blue line for the abundance of daphne in the water. And in Oneida Lake, which has a very reliable, every year there's always this clear water phase thing, you always have, it varies by the year. See, this year started out low, got high, and then went down again. This one, from right when they started measuring, it was already really high algae. This one started sort of at a moderate level and immediately started to decline. But in every case, the low algal abundance corresponds directly with the high Daphnia abundance. And that's because the Daphnia are filtering through the water and essentially mowing down, to forgive the metaphor. Those of us who work in aquatic systems think of filter feeding organisms like Daphnia as being basically analogous to cows or bison or something like that. They're the primary herbivore in the ecosystem. And so here's where the evolution comes in. When the algae are growing at this time of year, and there's not a lot of consumers there eating them, the ones that are growing the fastest and reproducing most often and growing and dividing most frequently are going to be the ones that rise in abundance. And the ones that grow more slowly, for whatever reason, are not going to be as common within a species and even among species. And when the Daphnia or some other consumer, it doesn't always have to be Daphnia, show up in abundance and start to graze out the phytoplankton or algae, they are not intentionally selected, like going through and picking out their favorite kind of food particle, but they're doing this filtering operation, and there's some things that are a lot more likely to get caught and consumed by Daphnia than others. So here's an image. This is not to scale, but I did want to at least demonstrate the fact that they're much bigger than their food. These are several different species of diatoms, which are algae, and they come in many different shapes and sizes. And definitely among all the different species, some of them are more likely to get eaten by Daphnia than others, because getting caught in all of these filtering appendages, which you can't really tell that they're appendages in this photo, but this is where all the action is. These antennae are used for swimming. Certain cells are much more likely to get caught than others. And so there's a dramatic change along this axis here. We see it as the water's not so clear, the water got really clear, and the water's not so clear again. But the algae that are there making the water not so clear here are dramatically different than the algae that are making the water not so clear here. And that's because of this selective effect of whatever's eating them. The ones that are most likely to get eaten are then underrepresented once they grow back. And the reason they go back, by the way, is because once the algae are at this low abundance, the Daphnia have run out of food, and they also crash, as you saw here. And so there's a selective effect that happens among the different algal species, which is not evolution, as we typically think of it. That's more of a community ecology, which species are present here and why, and it has to do with the grazing by Daphnia. But even within an individual species, as long as it is one that is able to persist through that period during which the Daphnia rise in abundance and take all those algal cells out. For any species that isn't completely driven temporarily extinct by the grazing pressure of the Daphnia, the cells that are left after all of that consumption that are then free to grow and divide and sort of repopulate the algal biomass are the ones that were least likely to get eaten. And so even within an individual species, there's a size selective effect where the longer cells that are slightly less likely to get pulled in by a Daphnia will be overrepresented after that clear water phase. And certainly the larger species are overrepresented. And so I had originally been thinking, well, at first I was thinking I would talk about the evolution that we see within freshwater communities that happens as a consequence of predation of zooplankton like Daphnia by fish. There's another whole scale of what all the different species of zooplankton that we see and how big they are and how long they are and whether they have a tail spine or not, that is all driven as an evolutionary process by the selective effect of visually hunting fish predators. But instead I decided to go one layer further down on the food web and to point out the sort of story that plays out every spring and summer in most north temperate lakes and ponds, which is this very strong, you can think about not only is the water getting clearer, but you can think about, I've given you an explanation for why that is, but also that what's going on during this dramatic decline is a huge selective event where certain species and certain sizes and shapes within a species of cells are being preferentially pulled out of the water, and others have phenotypes that are particularly well suited to avoiding consumption by something like Daphnia, and then those end up being overrepresented afterwards. And the reason that it plays out again the next year is that generally the particular species and individual types within species that are best suited to avoiding getting consumed by a grazer like Daphnia are not necessarily the ones that can grow the fastest when the water's still a little bit chilly, but there's nice long days of sunlight. And so when this all plays out again the following spring, even though they were the winners here, they actually don't end up being the winners here. And so there's this constant dynamic and interplay where certain types within species and certain species are being pulled out and brought to low abundance, and others are growing to high abundance. And it's all sort of going on underneath your canoe, and you don't even realize it. But this part you can see, and that can be a little cue to think about these sort of evolutionary dynamics that are occurring between when it's fish and zooplankton, we call it predator and prey. When it's Daphnia and algae, there's actually a bit of an argument about whether to call that predator and prey, because typically something that eats plants is not thought of as a predator. We think of that as an herbivore, a vegetarian type organism. But then if you get into the actual details of it, the predation relationship is usually defined in the ecology textbooks as when an organism consumes another organism whole, as opposed to taking bites out of it, like caterpillars do on leaves. And so arguments have been put forth that when Daphnia eat individual phytoplankton cells, which is the whole organism, that's actually a predator-prey relationship as well. I don't think the terms really matter, but I think the clear water phase is pretty cool, and you can watch for it this spring in your favorite lake or pond nearby. Thanks.
Questions for any of our distinguished company here?
Is Daphnia spending the winter hibernating in place, or?
That's a good question. It depends on the water body, but most zooplankton, including Daphnia, have a particular kind of egg that they lay at the end of their growing season that is known as a diapause egg. And it's a lot like a plant seed. It doesn't hatch right away, and it's very robust in the sense that it can be completely dried out, or it can be frozen, or it can be buried in the sediments, and then it's often those diapausing eggs that hatch out in the spring.
So in particular, Daphnia is just a one-year life cycle?
an individual organism, maximum lifespan for Daphnia is on the order of like a couple months. It depends a lot on water temperature. But certainly from the beginning of the summer to the end of the summer, there's been a complete turnover of individuals, and none of the same individuals are there at the end. But the way that they overwinter, you know, you guys, each of you mentioned ringwood, you know, many of those ponds dry out completely by late summer, and in a habitat like that, where the pond is not there year-round, and those diaposing eggs are essential for them to be able to come back again. In a bigger lake, they're able to overwinter under the ice even, but typically they're found at very low abundance. You know, if we go drill a hole in the ice and go pull nets, so you find hardly any of them, and so those diaposing eggs are their main mechanism for persisting through the winter, basically, so they can come back.
With an area that has so much citizen science for kids, and kids discover the trail with Museum of the Earth, and the Science Center, and the Lab of Ornithology, are there specific programs where the kids can learn to go look in their backyards and do their citizen science and report what they see? And,
well, I can speak to the lake side of that question, which is there's a pretty cool program known as the Floating Classroom.
Science Center.
Yeah, that
Floating classroom.
It's separate.
It's separate.
Oh,
yeah, it's not
I stand corrected.
part of the Science Center. I'm trying to remember what.
It's its own thing.
It's its own thing, right? Yeah, yeah. There's a I know a guy named Bill Foster who seems to be the guy who runs it too, pretty well. And that happens down on the. They have a boat that's docked there at the south end of the lake. I think that's a really great program. We're often actually. Bill Foster, the reason I know him is that we're often supplying him with laboratory-grown Daphnia so that he can do demonstrations for kids as part of that floating classroom program. But that's just one example. We basically have others. An answer to
It's,
that.
as usual, Gene, you asked a fantastic question, because one of the hidden reasons that we picked this theme this year is that PRI hopes to introduce exactly what you just asked about at the Nature Center in the coming couple of years, and so I've been feverishly taking notes. Other questions?
I have one for Jeremy. So the distribution of the Phoenicians and the Romans certainly colonized North Africa, but I didn't notice much there. Is that sampling bias or?
It is, but at that.
In Morocco, there was a couple of hits.
Yeah, no, there are actually other data available which fit with those Mediterranean lineages
Yeah.
in North Africa as well, yeah. But certainly, one of the studies we would like to do is to use mice to analyse Phoenician movements. There's been some very nice work on human DNA relating to Phoenician movements, basically getting samples from people around ancient settlements that could be related to the Phoenicians, like the present-day position, present-day city associated with Carthage. Yeah. And you know, there are certainly indications of particular genetic variants that are still present in those cities that seem to link those cities, and we feel we could use mice for in similar ways to sort of track where the Phoenicians were going.
So I have one for you too. What happens if you look in nuclear genes?
Yeah, it's very interesting. You don't get as strong a pattern. So it seems like the mitochondrial, this female difficulty for later colonizing females to displace the early colonizing seems to be much more female related thing. We particularly found it on the island of Madeira, which has got a very strong signal of a single colonisation, which was unexpected because we expect from northern Europe, which we relate to a Viking, probably a Viking colonisation, while the nuclear signal is very much Portugal, and it's completely different really, and much more genetically variable, more mice coming in. Seems to be the male side. Seems to be a male female difference in terms of colonisation.
That's really neat.
Question, looking at this map, I was thinking also of rats,
Yes.
and how rats were always taught were responsible for the Black Plague,
Yes.
etc.
Yes.
If data were collected on rats, would it show up basically similar patterns?
Certainly rat colonisation of Europe. So we're talking about, there's two sorts of rats. There's the black rat, the ship rat, which is Rattus rattus, and then there's the brown rat, the Norway rat, which is Rattus amphibians. Rattus rattus, I would expect might well have similarities to Thanos because it does seem that probably spread around Europe around similar sort of time. It was certainly was present in Britain at the Roman times. We know that. The Norway rat is a much more recent colonisation. That's 1700s is the earliest Norway rats in Europe. And they both black rat and brown rat seem to come from the Indian subcontinent, spread from there. So they seem to have spread from further afield than the house mice, which seem to be in the Near East colonisation. So I think the differences between them would be very interesting, actually.
Were rats and mice doing the Black Plague thing? Or was it
Mice
just
were
rats?
not, no.
Mice
I mean, even the rat thing, people are questioning, actually. But the traditional story is the black rat.
I have a question for all four of you, or any of you. So, in looking at the examples, and you guys were just totally perfect in your range of non-coordinated examples. So Willie talked about pedomorphic salamanders. Cole talked about slugs that get lungs and salamanders that lose lungs, and woodpeckers that come in different sizes and eat slightly different things. Jeremy talked about house mice in Europe. And Brooks talked about a very tiny time scale of annual changes in depth. Strikes me that some of these are stories about evolution, just change through time. And some of these stories are stories about natural selection. And so I wonder if any of you have any thoughts about, so Jeremy, you didn't mention natural selection. You just talked about change through time. And Willie, you didn't mention natural selection either, but change was kind of obviously implied. And Brooks mentioned almost nothing but natural selection for the diatoms, and Cole's woodpeckers are the kind of end result of natural selection. It's not kind of going on while you're watching. So if I was Joe, average person, and Josephine, average person, and I want to know how to see evolution in my backyard, is this all just the same thing? It's all evolution, or are there differences and similarities in just seeing evolutionary change versus seeing the mechanism of evolutionary change?
Well, so in the case of these salamanders in Mexico that keep their gills and live in water, one of the common explanations is that they metamorphose into a very dry terrestrial environment, which may not be such a great place to be a sapling.
On land.
On that, right. So that's one possible explanation for for why perennial-branchiate salamander lineages evolve multiple times. It does put a time scale on it potentially for you, as you look at the ages of these bodies of water. And knowing that makes some sort of idea, estimate about how long these lineages have existed. I think, you're looking for a selection event there. The selection event it's tough to beat. An epistematic salamander in a really dry terrestrial environment. Not that there aren't examples of that, it's just that it's tough. Why leave the water if you've got a nice situation there? Cool.
So, we talk to the students about evolution. Selection can happen every generation, because the kids often come in with this idea that they're thinking about macro patterns that they've seen, and it has to take a long time, geological time. And that's not true. That genes are reassorted every generation. And I think that as I was working up the kind of the woodpecker thing, I never really thought about that before. I've seen, traveling in Latin America, kingfishers, you go down the Amazon and there's like 6 different kingfishers that all do the same darn thing. They look pretty much the same. A kingfisher's a kingfisher's a kingfisher, but their bills are different and they can pull different sized fish out of the water. With the woodpeckers, we used to think that the Darwin's finches was a done deal, until Peter and Rosemary Grant started actually measuring bill length year after year after year with known birds, and they got a year with a really dry season, and the big birds that had the great nutcracker bills were able to handle the seeds from that dry year, and they reproduced differentially compared to the others, and the next year, bill length was bigger in the population. Now that didn't last because rains came and then the little ones came back, but you could see it on a year-to-year basis, and it made me wonder if anybody actually has looked at the downy hairy pair to see if there is some small change, fluctuating change maybe, but some change in that bill size that you could look at from year to year. And I'm just, I'm not an ornithologist, I don't play one on TV, but that makes me want to look to see in the literature if somebody has bothered to go look at that. So I think that there could be things on a small time scale that people could see. They're just not so dramatic. Like the stuff that Brooks mentioned about the, Daphnia getting spiny. It's unfortunately sort of. It's a more plastic kind of thing, right? In a fishy pond, they get spiny, but if you take the fish away, then they don't stay spiny.
Yes, but some species have much longer tail spines than others, in terms of how
Yeah.
long they can get.
Yeah. But
Yeah.
for somebody to go out and look at it, and maybe even do a backyard experiment, it's tough.
Your question is a good one, Warren, because it's about kind of the difference between the products of evolution by natural selection, the outcomes, or the actual process by which it occurs. And it's generally much easier to observe the products because we can observe them all now and see the different salamanders and woodpeckers, etc. And I've heard it said that evolutionary biology is essentially a historical science because, for the most part, we're making inferences about things that have happened in the past using a whole variety of tools in the present. But I also think that, particularly in the time that I've been doing this within the last 10 or 15 years, there's been an increasing appreciation for, in certain ecosystems with certain organisms, the process of evolution by natural selection actually happens fast enough that you can basically watch it happen if you're looking closely enough. But you have to be looking pretty closely. You have to be catching woodpeckers and measuring their bill depth, or you have to be taking plankton samples or something like that. And so the more obvious things to be able to go out and observe on your own, you backyard are the products rather than the process. But it's also fun to observe the process as well. Something that I thought of, well, when you mentioned it, Cole, that I could have done my whole 10 minutes about that, was you mentioned the goldenrod galls. That is a perfect backyard example because there's goldenrod all over the area. And so you'll often see, if you look closely, you get the stem of the goldenrod. These are these big leafy green things that are all over, like abandoned farmland or whatever. And you'll see this thing called a gall, which is this sort of bulbous formation in the stem. And that's because an insect laid an egg at the apical meristem as the plant was growing. And the egg hatches and the larva actually tricks the plant into changing expression of genes in order to form this little house for the insect larva to live in. So the gall doesn't come from the plant trying to fight off the insect. It comes from the insect manipulating the plant in order to create this little house. And I sat there trying to remember the name of the author who studied these in great detail and then wrote so many papers on them, and wrote a whole book.
Abramson and Weiss.
It's Abramson, okay. I was thinking Abramoff, and I was like, no, that's like some political criminal. Abramson, yeah. And so what's really cool about this is that if the gall is pretty small, then a woodpecker can peck in and get to the larva in the center and eat it. And so there's selection favoring forming a really large gall. No, I have it backwards. So the woodpeckers are able, because of their beaks, they can hack open anything. But they tend to see the biggest ones because they're most obvious. So if you make a gall that's really big, it's a nice big house for the insect, but it's most likely to get consumed by woodpeckers. However, if the gall is too small, there's a parasitoid wasp that has a little injector and lays its eggs inside the larva of the insect that's in there. And so there's actually selection pushing on both sides, and it depends on what kind of winter it is in terms of how strong the selection by the woodpeckers is. So if it's a really harsh winter and the woodpeckers eat a ton of these, that pushes towards those individuals that cause smaller galls. But then if you get a different kind of year and it's particularly good for these parasitoid wasps that can then inject and parasitize them, it sort of goes back and forth from year to year in terms of what the average gall size is, and it's sort of very similar to the way the Grant saw the beak size sort of going back and forth, and there's these sort of so we tend to think of evolution as having this divergent, and then you have a different endpoint than where you started, but there's all this sort of back and forth that goes on depending on what things were like from one to the next. It was a very long answer to your question. But because you mentioned starting programs at the Nature Center, I thought these galls would be
doable.
Perfect, yeah.
And I'll say this seems like it's a bad winter, obviously. It's going up. But I did a science cabaret in the first week of December talking about winter adaptation. And I wanted to bring a bunch of galls in to show them to people. So I went out in my backyard and was pulling the things. And I had to get to about 30 before I found the first one that hadn't been hit by a woodpecker. So I could actually find some maggots in there to show folks. I mean, they were just hammered. But it's a fun, it's a dial calipers kind of exercise that you can do with kids.
And it's also fun to cut them open with students and see the little maggot in
yeah,
there. Whoa.
And they're tasty.
Any other questions or discussion?
Okay, well, thank you.
All right, well, thank you all.